Welcome. We're going to be talking about risk assessment. So we've been spending a lot of time talking about these different core ideas. So that could be related to toxicology, that could be related to air quality or water quality or food safety or radiation. But this is one of the first times we're really going to be hammering on one of the practices of environmental health. And this is where risk assessment is and comes in. So what is it? How is it done? And how can we use risk assessments to identify vulnerable populations? So these slides came from Tanya Peterson, who uses risk assessments all the time, and I've adapted her slides just to kind of give you a sense of this scope related to risk assessment. This is likely more detailed than you're going to need, but just to give you a sense of, well, what are we talking about when we're thinking about risk assessment? So the short version is that it's a stepwise process of organizing information. So it's essentially a way of creating a report about the things that are happening. So what are the potential adverse effects? What are the dose levels where we might expect these adverse effects? And then who might be some of these populations of concern? So the reason that we do this is it informs decisions about the management of risks. This is the key point here. It does not define what is an acceptable risk. So as we're going through, so there are about 5 steps related to this. We're going to go through each one. Each of them are in different color-coded slides just to kind of give you a sense, and we can kind of keep track of where we are. So we're going to go into problem formation first. So what is the problem related to it? And then we're going to talk about what the hazards are, the dose response, and then what some of the exposures might be, and then characterizing risk. So as we go through each of these steps, we'll kind of highlight what we're talking about. So we're going to start with planning and problem formation. Okay, so this is all about who, what, and where is at risk, where is the hazard of concern, where does it come from, what's the exposure, and then it can go into some additional depth related to these questions. So for example, how long does it take for a hazard to cause a toxic effect? Does it matter when in a lifetime exposure? So we talked a little bit about, children are not small adults. And so as we're kind of thinking about that, that is going to make a difference. So some of the things that it really kind of considers is what are the major factors that are considered in risk assessment, and what is the context, what are the goals, the scope related to that. So ultimately, it involves a whole bunch of people in this process, but with a goal of producing some products. So one, it wants some sort of model that identifies what the sources are, what the stressors are, what the populations are, and then what's the analysis plan for the risk assessment. So we're going to be talking a little bit more about that. So the hazard assessment, the pathway analysis, so how is it getting into your body, for example? Where is it coming from? Is it coming from air or soil or water? And then we talked in Toxicology Week a lot about how these different environmental products, for example, can potentially bind to some of these receptors that are in our bodies to have health impacts. And so this is where this comes in. So what are the sensitive receptors that may be one of these roots, for example? Okay, so that was the problem formation in broad brushstrokes. So as we're thinking about the hazard identification, let's go into a little bit more related to that. So again, now we're in green. So this is all about hazard identification in these green slides. Okay, so as we're thinking about hazard identification, this is the process of whether an exposure to some sort of stressor can increase the incidence of pick an adverse health effect. It could be cancer, it could be birth defects, whatever those are. So what are those health effects and what are the circumstances in which these may be likely to occur? The data sources these can come from can be very broad. So it could be clinical studies, but they're kind of rare. You have epidemiological studies, animal studies, and then you have in vitro, so that's all cell culture, and then you can use computer models as well. So as we're going through, you have lots of data sources and you're trying to really kind of hammer at what are we expecting to see related to some of these hazards. There are 2 things that they generally will think of. One is these toxicokinetics. So this is the passage of that toxic agent and its metabolites through the body. And there's also these toxicodynamics. So these are the effects that these chemicals have on the human body. Other things that go into hazard identification is the weight of the evidence. So we've talked about this previously in other contexts, but what is this weight? What is the confidence of this actually thought to exist? So there's a suggestive evidence of carcinogenic potential, for example. hazard identification also includes the mode of action. And so this is especially important related to carcinogens. And so we're going to talk a little bit more about that as we get a little further. But ultimately, we want to evaluate the adequacy of the body of evidence to understand if a substance is posing a hazard to human health. Okay, as we get into dose response. So we're now going to dig a little bit more into this dose-response relationship. We talked a lot about this during Toxicology Week, so I don't think it's going to be any surprise that, the shape of the relationship is going to depend on, not only the toxic agent, but also the dose that is being delivered, for example, or that you're being exposed to, for example. In this risk assessment, though, there's so many possible relationships. So one of the issues is that toxicity research typically focuses on a limited number of adverse effects. We can't study everything. There sometimes aren't enough studies related to study everything. So again, there's a scope related to some of these risk assessment studies in dose response. The thing that I want you to kind of keep in mind is that the response that occurs at the lowest dose is generally selected as the critical effect for risk assessment. So it's assumed that, if the critical effect is prevented from occurring, then theoretically no other effects of concern should occur. So again, one limitation is if you're only doing a limited number of possible effects, then you may not know. But again, this is the underlying assumption. So as we're talking about this, the limitations is that when we're talking about this dose response data from humans, these data are often lacking. So we often move to animal studies as a way to increase the amount of available data. There are some issues related to, dose impacts. And so we're going to talk a lot about that, and it increases in uncertainty. So there are uncertainty factors that are kind of built into some of these considerations. Okay, so that was related to dose in a very broad nutshell. Let's dive in a little bit more into exposure assessment and kind of what that means. So these are all going to be in this pinkish brownish color. And so as we're kind of thinking about this, so exposure assessment is that process of measuring or estimating the magnitude, the frequency, and the duration of how humans are exposed to a certain environmental agent. So this could be now, this could be in the future. And the things to keep in mind related to this are, this could be the size, nature, and types of human populations that are exposed. So like how many individuals are getting exposed? What is the nature of that exposure? And what types of various situations are occurring? So exposure assessment will take into account the exposure pathway. So how is that agent coming from its source to the person, and what is that exposure route, meaning how is it getting into the body? And we talked a lot in previous weeks about, concentration, exposure, and dose. This is where this comes into play. And so what is the intake as it's, coming through a body opening, for example, and what is the uptake if it's being absorbed? So we talk a lot about, benzene related, when you have that ring structure, that phenolic ring structure, you may expect to see it being taken up through the skin potentially more frequently than a water-based compound, for example. When we're talking about some of these routes of exposure and how things are getting in, so this is a nice way of thinking about common exposure pathways. So related to air or soil or water, food, and how it can get into our bodies for an internal dose. So when we think about this range of exposure, so for any specific agent or site, there's going to be a range of exposures that people tend to experience. So you might expect a high degree of contact for, factory workers for an extended period of time because they're on the job, and you may expect a lower degree of contact for a shorter period for individuals who are using a recreational site downwind. So often related to exposure assessment, they will look for occupational exposures because these individuals may have an increased exposure, for example. They also quantify the exposure. So how are individuals actually being exposed? So you can look at these point of contact measurements. So this is exposure measured at the point of contact, so that outer boundary of the body. You can also look at exposure that's being related to scenario evaluation, exposure that's estimated by separately evaluating the exposure concentration and the time of contact and putting that two together. And then reconstruction is exposure estimated from a specific dose, for example. So then you can start looking at internal indicators like biomarkers and excretion related to that. So you're kind of putting all of these bits together. Okay, so as we start characterizing risk, this is going to be this next set. So this is as we use all of this information. So we use the results of these analyses to estimate the risk posed to ecological entities. And so that may include the degree of confidence in risk estimates. We can summarize uncertainties, we can cite additional evidence, and then we're starting to interpret what some of these issues are going to have, for example. So things we might keep in mind is this risk going to be acute or chronic? And what are the severity? And over what time might we expect this to happen? And so what might be the risk to not only humans, but also potentially other species as well? So, and if there are additional organisms at risk, we need to know that. So as we're starting to think about this, so then what is the nature and the presence or absence of these risks? So this is like that overall, as we're starting to interpret some of these things, what is the information about how these risks were assessed? What are the some assumptions and the things that we still aren't really aren't clear about? And then where are our policy choices kind of overlapping and will need to be made? So this overall final risk characterization component, this puts together all of these individual risk characterizations plus this integrative analysis of putting it all into perspective. It's really important. Okay, so this includes two stages. So one is the analysis of risk, and then the other is how do we communicate that risk related to these different problem formations and putting it all together. Okay, so if we're talking about the analysis of risk characterization, we can use some different terminology. So for example, margin of safety, and we're going to go into that a little bit. We can look at analysis of various chemical interactions. We can look at fractional responses. We can look at uncertainty. And we'll go through a little bit more about what each of those mean. So if we're thinking about margin of safety, and we kind of covered a little bit about this in Toxicology Week, but I'm not expecting you to necessarily remember it per se. However, as we're going through, margin of safety allows us to understand, approximate thresholds so we can reduce potential exposures to a population. So the threshold dose over the actual dose. And so the goal is that if you have a margin of safety that's over 1, you're not expecting to see a toxic effect related to this. So we're going to go through an example of what that means. So let's use this example. So based on experiments in mice, the human threshold dose for a specific solvent is estimated to be 21 milligrams per kilogram body weight per day. So that is the dose before we generally see an effect, for example, like a negative threshold effect. When we look at assessment of an exposure in a contaminated groundwater, for example, that exposure may be, so here's the maximum exposure level, we find that it's 0.672 mg per kg per day. So we are going to take both of these numbers and put it into a formula to understand margin of safety. Okay, so given those two, what might be that margin of safety at this level of exposure? So to calculate this, what we'll do is we will take that threshold dose, that 21 mg per kg per day, divided by what that exposure is of 0.672 mg per kg per day to give a number, a margin of safety number of 31. So again, anything over than 1 is considered favorable in terms of having a higher margin of safety before you start to see an effect. So we could increase the exposure by a factor of 30 before toxic effects would be expected to occur, just to kind of give you some scope of how this works. The downside or the issues, the limitations is it's really hard to estimate what a threshold dose is. So data are rarely available. So we start to use these safety factors to start to estimate what a threshold dose may be. And based on all of these modified thresholds, there's an acceptable daily intake. And that's, if you're below that, you shouldn't expect to see adverse health effects, or they would be expected to be infrequent. So when we have these adjusted margins of safety, for example, that's usually this threshold dose, this estimate of threshold dose over the actual dose times a bunch of old safety factors. So just this gives us that uncertainty. So we have a conservative estimate of that margin of safety rather than something that has, so we want something to have that has plenty of wiggle room and uncertainty built into that. Okay, and what might that look like? So some of these reference doses and uncertainty factors. So reference dose, so this is that conservative measure. So what is the dose that would be expected that you could be exposed to without an appreciable effect over a lifetime of exposure for the most sensitive member of a population? So this is that reference dose, that lowest dose. And to find that reference dose, you use this no observable adverse effects level, so NOAEL for short, over a bunch of uncertainty factors. And those uncertainty factors have a lot of, there are many, and we're going to talk about that. But it accounts for the variability and sensitivity among populations and extrapolating between animals and humans, for example. Reference concentration. This is the same thing as reference dose, but except that it's references concentration instead of a dose. So because NOAELs are really difficult to determine and extrapolate, so often benchmark doses are used instead. So benchmark doses, that's the dose that corresponds to a specific level of response. We talked a lot about this in toxicology related to effective dose 50 or lethal dose 50, LD 50, or toxic dose 50, TD 50. But if you bring that dose down to a percentage of a response, so you can do lethal dose for 1% of a population, that's LD1, for example, or toxic dose for 1% of the population, TD1, and you can start to look at some of these, estimate some of these benchmark doses. So it kind of gives you some sort of dose response function where you can start to place some of this data into perspective. So the benchmark dose lower limit, that's the dose that corresponds to a specific level of a response, and it is often used to determine the reference dose. The reference dose, again, is the dose that you can take over the lifetime of exposure without any expected negative effects. So this kind of, you can see how it starts to approximate and how we're starting to estimate what some of these, what these factors may be. So as you're thinking about uncertainty factors, know that you can go to the EPA's Integrated Risk Information System, or IRIS for short. They have a lot of information on these various uncertainty factors for various chemicals, and it can give you some context for kind of information for using in your risk assessments. And also know that these uncertainty factors give us a lot of wiggle room, so we can be more conservative in understanding what a dose that is not expected to have impacts may be. So if we have a lot of uncertainty factors built in and it's quite large, you can assume that we probably don't have enough information necessarily to really derive a solid reference value. In terms of some other terms that you might hear, so there are these hazard quotients, for example. So a hazard quotient is the dose over the reference dose, or the concentration over the reference concentration. So again, what is the dose that or the concentration that's happening over the dose or concentration that it's not expected to have an adverse effect? So if you have a hazard quotient that's less than 1, you would expect that you probably are not going to have any adverse effects. But if you're above 1, that is indicative of concern because it is over the level of expected to have. So again, that reference dose is the dose that you can have before in a lifetime before having any appreciable negative effects, theoretically. We also have what's called a hazard index. So this is quantifying risk from exposure to multiple contaminants because none of these are in isolation. So this is the sum of all of those hazard quotients into a single hazard index. So again, if the hazard index is less than 1 from all of these different things, you can probably expect that no negative effects are likely to be observed. But if you have a hazard quotient that's, or a hazard index that's above 1, you might need some additional judgment to really interpret the effects of these multiple chemical exposures. So as we're kind of talking and thinking about these, what might some of those be? So hazard quotients, so that could be the ratio of a chemical contaminant concentration to a selected screening benchmark. And some of these parameters could be, the area that we're talking about. So what is the range that is being used? It could be a home, for example. It could be a work site. It could be related. Next one is the food ingestion rate. So how much food is typically being consumed over a specific day? What is the bioaccumulation rate? So if we're talking about persistent organic pollutants, for example, we know that those bioaccumulate. So that process may be a little bit different as you're kind of considering these hazard indices. Bioavailability is another thing to consider. So we talked a lot about this related to lead. So how bioavailable is the compound that we're talking about? And then what is the life stage of the individual? So if we're talking about, lead in a young child, this is going to be way more impactful than if we're talking about, an elderly individual exposed to lead. We don't want lead anytime, but if we're talking about young children, this should signal to the back of your brain like, oh, I really need to pay attention to this. We talked a little bit about chemical mixtures previously. So you can have three possible effects. So these can be additive, where if you add A plus B, you have a linear increase. So one is 3, one's 2, you get an increase of 5, for example. But synergistic toxicity, we saw that with the mixed metals through the Robert Wright podcast. So synergistic, you could have 3 plus 2, and you don't get 5, you get 25, because those two work together to produce an effect that is way bigger than either of those together. And then sometimes you'll have these antagonistic effects where one might antagonize the other when they're added one at a time. So keep these in mind. There's also fractional responses. So we take into account these margin of safety exposures, but as we're thinking about carcinogens, they don't quite work that way. So this instead is these fractional responses for estimating dose and then what fraction of the population could be affected using a dose response model. So know that it can get really complicated as you're starting to think about this, but ultimately, what's the population responding? That is dependent on what the cancer slope factor is, which is something that you can look up, and the chemical dose. And again, there are formulas for all of this. So know that if you want to go down the rabbit hole for how this works, you can totally do that, and people do. So this is just an example for calculating some of these, where you can start to look at PCBs at a specific dose and put them together with a cancer slope factor. So what might this look like, when you start to look at these additive impacts? And again, I'm not going to make you do this, but know that people do this all the time. So if 10,000 people are exposed at a certain level, the upper bound of a specific number of contaminant-induced cancers, you can calculate that. So you can see you can get into the weeds very, very quickly. So I want to kind of pull back related to that, but the overall goals related to some of these risk characterizations is being really transparent about what you would expect, what are the assumptions, what's the logic that you're using, what are the limitations. And then you also want to be really clear. So the products of risk assessment should be understood by readers who are not only scientists, but individuals who are outside of that risk assessment process. So you can imagine that, as we start to get into some of these reports, there's a ton of jargon, there's a ton of terminology, and it can be really confusing for someone from the lay public to read. So how can we make these better so individuals all across our country can better understand them? As we have these risk characterizations, we want to be consistent. So between EPA policy and other programs, and as we're kind of going through, is one person doing it in a similar way as a scientist over here versus over there, versus, across states. So how can we kind of pull these together to be similar in scope? And then we want to be reasonable based on sound judgment and methods and limitations and, the state of the science. So all of these things are pulled into risk characterization. Okay, so that was that risk characterization. Finally, we have this risk management. And this is finally that decision-making step. So we pull everything together and we have all of that scientific data. And at this stage, this is where we also bring in these non-science factors. So what's the acceptable level of risk? What's the existing legislation? What are the costs of doing it? How easy is it to implement? What are the stakeholder perceptions? What are the considerations related to administration for doing it? All of these things are going to matter. So as we think about all of this, always consider environmental justice in the things that you're doing, not only in risk assessment, but these different exposures that we're talking about. You can imagine that you can use risk assessment as a way to kind of think about these environmental injustices and inequitable exposures, and how can we do better? So that is where I'd love for you to focus for moving forward.